This video I will show you how we do the cell culture. So this is a typical tissue culture flask where we put our cells inside this. So right now I'm going to culture one cell. I'm not going to name it, but in general we are doing some HeLa cell culture. So this is how the cells looks like when after centrifugation. They have gone down based on their density. After centrifugation, I just did like 1000 G force in the centrifugation machine. So what we generally do is, for example, we have something called trypsin enzyme, which what it does is it's an enzyme which detaches your cells from the surface. And then we have something called the complete medium. So each of the cells has its own complete medium. For example, typically we use DMEM, Dolbeco modified essential medium, or there are something EMEM. Depending on the cell types or depending on the cells, you have to use different kind of complete medium. So you have to add DMEM or with its protein like FBS, fetal bovine serum, FBS, and also you have to add penicillin streptomycin, which is an antibiotic. So all these are specific to a particular cell type. It's not like so I'm just talking, for example, I'm just talking about HeLa cell. So for HeLa cell, generally we use DMEM plus 10% FBS, or some people even use 15 to 20% FBS depending on their requirement and experimental condition, and they also add 1% penicillin streptomycin. So what we do first of all, what we do is we for example, if you are taking out cells directly from the nitrogen tank, then you have to first thaw using the water bath, which I just showed you, and then we take it inside a falcon tube, that is around 15 ml falcon tube, and then you have to add the complete medium, which I just showed you here. It looks like something like this, and after that you have to maintain its volume, and you have to balance out by using another falcon tube similar like this, and then after that you have to centrifuge around 1000 G force. So different labs have different kind of protocol. So somebody used 1000, somebody 800, and also, for example, in general, those cells which are very sensitive, they generally, for that we use very less G force, like 800 or 500 G force. And once the cells get settled down here, then we have to add again this. We have to take out those supernatant and throw, and then we have to add this complete medium in it, and then we have to resuspend using this cell aspirator, and by using this aspirator here, and then you have to add it in your this falcon tube, and then you can put it inside your CO2 chamber, and then it can the cells can start grow. They will start growing and they will proliferate, and that's how we do the first step of cell culture, like if we are going to take out directly from the nitrogen tank. And in the next I will tell you about how we propagate them. For example, if you have to propagate them from one plate to another plate, you have to do like multiple plates. Then you have to trypsinize using that enzyme called trypsin, and you have, after trypsinization, they will start floating, and you have to again add the complete medium. And after so that is called trypsinization, and then the subsequent step is called detrypsinization. And after detrypsinization, you can depending on your requirement, you can replate those cells in another different plates, and that's how you do the replating, or you call it as a splitting of your cells. So thank you.